First of all, thank you organizers for allowing me to talk on the role of protein quality control systems in alpha-synucleopathies. I'm Harm Kampinga from Groningen, the Netherlands, and my work has been concentrated over the last few years on protein quality control systems and how they deal with protein misfolding diseases. Now, alpha-synucleopathies are protein aggregation diseases, in my view, in which amyloids are formed that are considered to have prion-like properties. So in post-mortem brain, as we all probably know, we have these Lewy bodies consisting of alpha-synuclein that reside in the brain cells. And there is this specific pattern of spreading throughout the brain of the disease with the related symptoms that seems to coincide with the movement of these aggregates from cells to cells in a spreading-like manner. Now, the question that has been often asked is this aggregation actually driving the disease at all? And what actually controls protein folding and hence prevents aggregation if this is important? And why does all of this happen mostly only late in life? And of course, the final thing, can we do something about it? Can we prevent or reverse it? Now, first, this question that I don't want to dwell on very long is this protein aggregation driving disease at all? It has been asked for many of these diseases, and my answer would be yes. And the evidence is actually multifold. I'm just showing you two features. First of all, alpha-synuclein, the key protein in Parkinson's disease and alpha-synucleopathies, has unstructured features and a core, a NAC core, that has amyloidogenic properties. Now, disease-related mutations that are associated with familiar forms of the disease with an earlier onset, they actually enhance the aggregation propensity of these alpha-synuclein proteins, or they have a decreased turnover rate, so the protein accumulates more. And so the protein, either through a lipid-bound mechanism or to a condensation mechanism, can actually undergo this process of forming a prefibrillar to a fibrillar structure and then forming these inclusions. And so without giving you much more evidence, I think the idea that this is driving diseases has been very clear. And so the next question is, what normally controls folding of proteins and would prevent such an aggregation? Now, this is, in fact, what is being asked in a consortium that I'm part of, that is funded by JMPD and spearheaded by Carmen Nussbaum from Heidelberg, and that also involves Bernd Buchau, Ronald Melky, Christian Hansen, and myself. And what we've asked as a question is, what is the role of protein quality control in these systems, and especially the role of chaperones? And then in three phases, what actually causes the initial aggregation in a cell, unseeded, without any extracellular material being present yet? And then what can these chaperones play? What role can they play in the propagation of alpha-synuclein aggregation in what we refer to as seeded aggregation? And then, is there a possibility of the chaperones breaking down fibrils and being prevented in these diseases in such a manner? So, first of all, let me talk a little bit with you about protein quality control mechanisms. So on the left side, you see what I actually like to refer to as protein homeostasis. Protein homeostasis being the balance between the proteins that we make, transport, and assemble, and timely degrade, and how the quality control system actually is involved in making sure that this is happening right. So for every protein that is being synthesized, it needs to be folded into folding intermediates and in the end to the native state. But in the meantime, in that process, you can actually get misfolded states or even misfolding happening from the native state that then has the risk of forming protein aggregation. And at any given level of this folding process, there is reversibility. That means that there is systems that can actually go back to the other states and make sure that this aggregation is prevented. On the other side of the spectrum, you have protein quality control systems like the ubiquitin proteasomal system, as well as the autophagosomal system, that can take care of these misfolded or aggregated species, remove them, or that also can take care of normal native proteins in a normal turnover process. And this entire system we call protein homeostasis, and it is regulated, as I said, by protein quality control mechanisms. Now, the systems that we're interested in is what we call the chaperone network. And that you see here on the right hand, this is a intricate network of all kinds of molecular chaperones of different families and different numbers in these families that take care of recognizing these specific species, these intermediate species, and then know what to do and how to communicate those to be either degraded, to disaggregate them, or to fold them into the correct strains. So this chaperone network is what we're specifically involved in. So let me first now go back and ask the question. These diseases, including alpha-synucleinopathies, are actually a set of diseases that all happen mostly lately in life. And Parkinson, in that sense, is one of the diseases that we see this with. There's Alzheimer's disease, polyglutamine diseases, prion diseases, ALS, and several myopathies, and that are all characterized by this protein aggregation and nearly always are late onset. And so what is the model that we use to sort of try and understand this? So what we do know is that with aging, this protein quality control system, the green curve here, actually declines when we get older. At the same time, just as a natural process, for instance, due to somatic mutations, we accumulate more and more protein damage burden during our lives, which needs extra handling of this protein quality control network. And at the virtue of where this burden exceeds the capacity, we have collapse of protein homeostasis, and we get, in this model, the non-genetic forms of many of these diseases. However, there's genetic forms of these diseases, in particular one that we've been interested in the beginning very much, and this is polyglutamine diseases, for instance. They're entirely genetic, dominantly inherited diseases where there is a polyglutamine expansion that causes the protein to be aggregation prone. This, however, is true from the beginning of life. So what basically happens with this genetic mutation, that particular person at the beginning of life starts with this extra protein damage burden, but because of the capacity being so high, nothing happens. And in fact, these patients get born normally and have no problems during their early adulthood. However, because of this increased burden, they have a more early collapse of protein homeostasis, and therefore the genetic forms of these diseases, they usually are seen very early onset, or earlier onset in life than the non-genetic versions. So with this disease, we started to use them because it's so easy to understand, completely genetic. We asked the question, can we do something about it to prevent or reverse it? And we turned to this protein quality control network, or especially the chaperone network, where we focused on the HSP70 cycle, which is actually depicted here, and in particularly on the J proteins. These J proteins, or JDP, J domain containing proteins, they are usually the ones that recognizes the misfold in the cells, or the fold in the cells of the proteins, that are then next put into these machineries of networks for them to handle them. And there is over 50 different ones of them, with the idea that some of them may have specificity for certain types of folds or even proteins. And so the question that we ask is, can we identify members within this family that are rate limiting, in this case, in suppressing polyglutamine Huntington? And in the next slide, I'm actually summarizing 12 years of work, because this is, of course, only on Huntington, but I'm showing you the discovery of this one DNA J protein called DNA JB6 that was found to be a rate-limiting chaperone in the onset of unseeded polyglutamine aggregation toxicity and disease. So here, first of all, you see the in vitro work that we did, and we look here at the formation of amyloids by the expanded polyglutamine protein, and it goes very rapidly. Just the addition of 1 over 100 molecules of DNA JB6 can already delay that onset of aggregation dramatically in vitro, and at a 1:10 ratio, we see a substantial delay in the formation of these aggregates. Also in cells, we can look at these puncta reflecting polyglutamine aggregations, and then when we put in different kind of chaperones, none of them actually changes the appearance of these punctae except for DNA JB6, and its testic-specific ortholog DNA JB8. Also biochemically, you can detect it. This is a filter trap assay where we actually trap the aggregates in undiluted, 5-fold diluted and 25-fold diluted samples of the cells that we express these proteins in. Without the chaperone, everything aggregates, and with these various chaperones, mostly aggregation is not changed except for the DNA JB6. Also, when you look at neurons and you put a polyglutamine protein into a neuron, what you see is that these neurites sort of disappear. You see that in this quantification here. DNA JB6 in unstressed cells doesn't do anything on neurite outgrowth. However, in case of co-expression with the polyQ, it completely inhibits the reduction in neurites and processes in these neurons. So it's very protective. It doesn't only protect against the aggregation, but also against disease, if you like. And then when we express this protein, for instance, in Drosophila models or mouse models of Huntington's disease, we can see that the onset of death is delayed by the co-expression of the DNA JB6 protein. So also in vivo, it delays not only the aggregation, but also the disease onset. So coming back to the model, the polyglutamine Huntington increases the damage burden if we give the cells capacity to actually counteract the aggregation by DNA JB6. We change the curve downwards, and therefore we delay the onset of the disease. And obviously now the question is, can we do the same thing for instance, Parkinson's disease, in which we also know that there are certain mutations and amplifications that actually predispose people for genetic forms of these diseases with an earlier onset. And so this DNA JB6 that we actually had discovered was next discovered to be very broadly anti-amyloidogenic. And one of these evidences is then can indeed that chaperone or other chaperones also block the initiation of that aggregation. So in the first question in our JP&D project was, can chaperones delay the initiation of unseeded aggregation of alpha-synuclein? And one of our partners actually showed that it could be the case. This was done by Chris Hansen's group. And what you're looking at here is, again, such a theoflavin assay for the amyloid formation of, in this case now, alpha-synuclein. And you can see that HSP70 doesn't really change that interaction, but DNA JB6 reduces that particular amyloid formation significantly. And if you then add in HSP70, it actually doesn't help. So it's really this guy, DNA JB6, and also for alpha-synuclein, seems to be a rate-limiting effect. So it delays the onset of the aggregation. Now in vitro, it was very, in cells, I mean, it was much more difficult to actually look at that. And so what Chris did and his group, they actually made a knockout of DNA JB6 and then scored for alpha-syn, here in red, punctate in cells. And whereas the wild-type cells didn't show much of punctate, what he could find is that the knockout of DNA JB6 increased the appearance of punctate in these cells. Now, if you reintroduce DNA JB6 into these knockout clones, you again go back to background levels of aggregation. So also in cells, more alpha-synuclein aggregation is seen without DNA JB6. So that was very encouraging. And then we also collaborated with him on this. So we made knockout lines ourselves and showed that in these knockout lines, and now you have to look here at this particular pellet fraction here, in the DNA JB6 knockout lines, there was more aggregates, more material found than in the wild-type cells, suggesting indeed that the presence of DNA JB6 is very important for cells to be sensitive to the aggregation. However, we were not so happy with these data per se, because in most of the cells that we have, DNA JB6 is actually expressed, and we saw very little aggregation, as you can see here, in cells that had DNA JB6. And we don't want to enhance the situation, make it worse, but we want to improve it. So we decided to set up an entire new system for alpha-synuclein aggregation, and this was done by a talented postdoc in the lab, Eduardo Matos, in which he fused a GFP to the alpha-synuclein, either a wild type or a mutant form that causes early onset disease. And then he made stable lines of them. in which he could, by a tetracycline treatment, turn on the alpha-synuclein protein that then would be green cells, and should look at puncta in live cell imaging, either in the presence or absence of added chaperones. And here you see one of those immunofluorescence images, and you see indeed also now, like for the polyQ, we seem to get puncta in these cells, and the wild type actually shows less than the mutant, very consistent with our sort of initial hypothesis. However, then things started to be a little bit more fuzzy, because when we did the fractionation types of experiments and looked at pellets for aggregate formation, there was not much of a difference between the wild type and the mutant form of alpha-synuclein, despite the fact that we got more puncta, and this is actually quantified here. So that was a first worry. And again, we wanted to do this now with asking the question whether or not chaperones could reduce this aggregation. So we decided to do another screen, but then first with the wild type synuclein first, because that's where most of the cases are actually associated with. And so this is how this then looks like. You have your chaperones co-expressed as indicated here, and then you look at your fractionated material, and of course we then quantify the pellet. And already you can immediately probably appreciate that not much was going on. And so if we quantify this, in fact, what we found was a very disappointing result. Without any chaperones, this is the 100% of aggregation that we measure. And then what the individual chaperones were doing can actually be depicted by these various bar graphs. And although we have some chaperones like DNA JB2 that showed some of effect, the chaperone that we actually were so much interested in and had been proven, of which it had been proven that it could inhibit amyloid formation of alpha-synuclein, had virtually no effect. So this made us start doubting about our entire system. So we went to our partner, Ronald Melke, who had built up this in vitro fibril amplification assay. And the basic thing what he does, he takes, for instance, material from healthy control brains or from PD brains, and he adds this to a signal, and if there is amyloids there, real alpha-synuclein amyloids, signal should be here, and the healthy control should be here. And without showing much data any further, what you can very much appreciate is that all our material that we sent to him as aggregated puncta insoluble material did not qualify as being a bona fide alpha-synuclein fibril. Now, this, of course, was a disappointment, but it meant to us that, in fact, as we already hinted in the beginning, is that unseeded alpha-synuclein aggregation in cells is not very easily happening. And it apparently requires other extracellular stress events that we haven't been able to discover in order for it to form real amyloid. And we're working on that very hard now to figure out what these could be. Also a warning to all of us who look at puncta and blobs in cells. Not all puncta are alike, and not all of them are actually bona fide amyloids, and therefore can be considered as something realistic for a disease. Now, we went, of course, one step further, and we said, okay, then let's start with some fibrils that we already have and ask the question whether or not we have chaperones or chaperones like DNA JB6 working in seeded aggregation. And again, it was Chris Hansen who has shown now that that indeed can be the case. So he again showed that the molecular chaperone DNA JB6, but not one of his family members DNA JB1, can suppress the seeded aggregation of alpha-synuclein in cells. And again, this is very nice and clear work, but we're working further on this because what this was all based on is knockout lines. And as I told you before, we want to understand what could be the protective effects of upregulation of DNA JB6 or other chaperones on what we call endogenous aggregation in cells that have a normal chaperone network, because that's what we're actually very much interested in to make things better. So we're working on this. So the final question then that we ask in our consortium is there a role of chaperones in the breakdown of alpha-synuclein fibers? And in fact, before we started with the consortium, already Bernd Buchau had shown that indeed there is a complex that he called the HSP70 disaggregase complex that can reverse alpha-synuclein induced amyloid fibrils. And the evidence is actually shown here in this slide. So what he showed is that if you look at where HSPs can bind, there is this specific set of HSC70, DNA JB1, and APG2, or also called HSP 110, that can bind to the side chains in these cores of the amyloid fibrils. And in fact, if you bring in DNA JB1 together with HSP70, HSP70 gets concentrated at specific sites on the side of these fibrils. More importantly, this is what the input is. And this is what the system looks like when you have incubated them with the entire tree chaperones and ATP. You can see complete fibril fragmentation. And in fact, recently now, this has been investigated in much more detail by Ann Wenting, who's also part of our consortium, and she's now shown that how this works. So DNA JB1 actually binds to the sites of the core of the amyloid fiber. And what it does, it recruits basically HSP70 to these particular sites. The HSP 110, the other player in this reaction, makes sure that all of the HSP70 gets into a configuration, we call this the ATP configuration, so that it can actually accumulate on this fiber. And now the fiber breaks into pieces, and you generate you disaggregate or fragmentate the fiber, and you get these fragments. And the next question we ask, okay, that's great, but what about these fragments? Are these now seeding competent? And even, are they toxic? Can they actually contribute to the toxicity? And the answer is yes. If you, and this is published by the other partner of our consortium, Carmen Nussbaum, now together with Bernd Buchau, when you actually take this disaggregation system in C. elegans, it generates spreading competent toxic alpha-synuclein species. So this reaction really results in seeding competent and putative toxic species. Now the question is, why would one have such a disaggregation system if it would make things toxic? And the question is, we don't know. But the question is also, maybe there is systems but downstream of this fragmentation that can actually take care of it. And we have some evidence that there is actually downstream of this particular fragment now cellular protein quality control systems other than these chaperones that can handle these fragmented fibrils to detoxify them. And with that, I come to the conclusions of my talk. So as far as we now have seen, there is it's very hard to actually do and mimic unseeded aggregation in the initiation of alpha-synuclein aggregation in cells. And it's poorly understood what actually triggers that particular feature to trigger the disease, because, I mean, once it has to be triggered, right? Then what we have as data is that in terms of the propagation of alpha-synuclein aggregation, DNA JB6, the one protein that was very anti-amyloidogenic, seems to have a function here, but this requires further studying. With regards to the role of chaperones in the breakdown of alpha-synuclein fibers, yes, there is a different complex, not involving DNA JB6, but DNA JB1, that can, in fact, break down these fibers. And we need to understand what is the downstream handling of these potentially toxic seeds to get further insight in the beneficial reaction of that particular system. And with that, I would like to acknowledge the people that were involved in the work, the people for the GPND project. I already mentioned most of those. And these were the people that are involved in much of the polyglutamine and other DNA-J work that we've been doing with Eduardo Matos actually being the guy that also now built up the cellular systems for alpha-synuclein aggregation. And with that, I thank you for your attention.